So good evening, everyone. I am your host for tonight. My name is Atika, a Master Research Student in Pharmaceutical Technology at IIM. So this science chat is organized by MBIOS and media partnered by 100 scientists of Malaysia. For tonight's agenda, the presentation by the speaker will be roughly around 20 minutes on their research, and then we will proceed with a Q&A session. If you have any question for the speaker, please feel free to drop the questions in the chat box at any time. So for tonight, we have Danial Saifuddin as our speaker. He is a PhD candidate at University of Queensland and currently in his second year in pharmacology field. His current research involved in improving stability and efficacy of opioid peptides via amino acid substitution. I will let him introduce himself further. So over to you, Daniel.
Right, thank you, Atika. So yes, thank you for the brief introduction. So my name is Danial Saifuddin. I will be having a sharing session about my research and also my work of a PhD student, where I work on creating new cures for the opioid pandemic, essentially. So I'll just open up basically a brief story about myself. How did I end up here? So I'll just start with my secondary school. So I actually did my secondary school in a Chinese national school called SMJK Chongqing in Kota Baru. So surprisingly, I was also joined a lot of co-curriculum activities like Scouts and rose up to the ranks as being president. Then I move on to take my A-levels in Tunku Abdul Rahman College, where I did my pre-U and A-levels for my university. So here's also like a picture of my classmates. And then I went to the University of Queensland to do my Bachelor of Biomedical Science. So from there, I had a lot of research projects and opportunities. So my first research project was actually to deliver peptides and proteins using liposomes orally. So as we know that peptides are highly unstable in the GI tract, so we actually use stabilizers like nanoparticles to actually prevent it from being digested and absorbed. Then we move on to I moved on to another lab where we work on soluble gels, where it is a solution in room temperature and a gel once it hits body temperature. So this actually worked very well for like sustained delivery. And I'll briefly discuss that at the end of my talk. Then after that, when I graduated, I managed to land a job in Tech Dome Penang, whereby I was actually a science communicator and we work on multiple workshops, especially like for my case, my product was a candle clock, whereby we actually have candles that will burn in different times and each layer has a different scent. So it's like a kueh lapis, but instead we make it into a candle, and for like 2 hours you have lavender, next hour you have vanilla. So it became an interesting way to make kids interested in science. And that helped also me to actually get the next job, which is a Perdana Fellow, which is more or less an intern within the ministry. So I managed to get a position in the Ministry of Human Resources, whereby we help with improving labor laws and also like some work with the technical vocational school. Then after that, my research project. So my current research project is to work on novel opioid peptides, whereby this is basically an opioid receptor, and this is basically the opioid peptide that I work on. So I'll give a brief overview of pain just to give an introduction for everyone. So pain is an unpleasant sensation that is felt through the sensation of extreme heat, cold, pressure, or even like mechanical sensation. So basically when pain is felt, like this man who actually placed his feet under the fire, it will be transmitted through the mechanosensory receptors up to the spinal cord and up to the cortex, where the brain will process the stimuli, and this is where pain is processed and felt. and then it will send down a signal to move away from the pain. So how do we address pain, especially in the cases of chronic pain through disease like inflammation, and also through like chemotherapy, whereby the patient is always constantly feeling pain? So ideally, the WHO, the World Health Organization, would start with non-opioids such as paracetamol or ibuprofen. So this would just help ease the inflammation. But when it gets worse and requires stronger treatments, we would then start with weak opioids and also maybe a stronger non-NSAID, like anti-inflammatory agent, such as steroids or another drug. And then severe pain is where we start with strong opioids. So these are like basically a list of strong opioids and also the uses of it, as well as the side effects that we can see over here. So most of the side effects can be subdivided into peripheral and central, whereby the central side effects are the most problematic due to the experience of sedation, tolerance and dependence, and also addiction, whereby then the patient will actually start to request for more opioids while they're already more or less cured from the pain. And this is due to the effect of receptor internalization, which I'll explain a little bit more later. So I'll start with how opioids were discovered. So it actually was started in the Mesopotamian area, where they found the poppy plant, and they actually they smoke the poppy plant directly, and then also discovered that there are extracts where they make it into tinctures to be consumed orally. And this also created the whole addictive and euphoric effect, while at the same time helping with people who are suffering through pain. And then morphine was actually isolated from the poppy extract and found that this is the molecule that actually helps ease the pain. Then we had the problem was that there were morphine addiction, where people were still requesting more morphine. And so the US actually developed heroin in order to cure the morphine addiction. They did cure the morphine addiction, but they started a heroin addiction. And so drug companies started to make more and more small molecule drugs that are similar to morphine, but hoping that it will not trigger the side effects. So for example, Vicodin, I think if anyone who watched House would know how well addictive Vicodin is, and where you can see House actually just pop multiple Vicodin pills anytime he just wants that kick. So this actually created a problem in the society where everyone is more or less chained to this opioid prescription drug, and they would try and request more. And even sometimes there are even crimes that happen with opioids. Like I can just give an anecdotal scenario where my friend who's a pharmacist, her pharmacist was robbed just because they wanted to actually steal oxycodone from the pharmacy. And so these kind of crimes just created much more problems in society when it is just to cure one commonly felt sensation, which is pain. So let's dive in a little bit more with how pain works. So most opioids work on the opioid receptors, which can be divided into delta, mu, kappa, and also nociceptin. And these are also the small side effects that you can get from each of these opioid receptors. So, and we also have endogenous opioids, which you can see on the top over here, which we will elaborate more slightly later. So how do these receptors work? So within the synapse, where the pain signal is being transmitted up through the dorsal root ganglion to the central spine, there is GPCR signaling. So in the normal basal condition where the pain is felt, there will be the action potential passing through the presynaptic cleft, and there are opioid receptors present in both ends. So in the case of when there is no opioids, the calcium is taken in and cyclic AMP is secreted out by adenylyl cyclase to ensure that the action potential is passed through the synaptic cleft, and this is where the pain signal will pass through. So in order to curb the pain signal, opioids would be activating the opioid receptors, whereby it inhibits the adenylyl cyclase and also the calcium ion channel, and at the same time hyperpolarize the synaptic cleft, thus preventing the sensation of pain to be passed through to the central nervous system. At the same time, when opioid receptors are activated, it started the beta arrestin signaling. So this is what happens when the opioid receptor is activated. The opioid receptor is internalized into the cell directly, where it will go through two pathways, where it is recycled or it will be degraded. And this actually creates more what we call a tolerance, whereby there will be less receptors available on the surface of the cell, and we require more of the same drug to have the same inhibitory effect that we wish to achieve. So this is where the root cause of our opioid pandemic happens. And there are also other factors like G protein decoupling, but that will be discussed later in another time. So one fun fact is that our body actually have our own opioid, opioid production. So our immune cells actually secrete endogenous opioid peptides in the form of peptides when there is a inflammation happening within the skin. So when an inflammation happens, all of the inflammatory factors such as interleukin, and TNF alpha would actually attract the immune cells to come in and do its job. But at the same time, it actually secretes out opioid peptides, whereby it will then create the anti-nociceptive effect and reducing the pain. So this is actually, you can actually experience this before, with like a paper cut, whereby you will feel a slight pain. But once the immune cells come in, you won't feel the pain for a while, but wait till the next day and you will actually start feeling the inflammatory pain. And this is where it is actually very helpful in an evolutionary sense, whereby when a creature, so these opioid peptides are also found throughout different species, but as what we have observed is that when a person who is injured, so like imagine a soldier over here got shot, they are still able to not feel as much pain and enable themselves to run to safety in order for them to recover. So this is also, you will actually find that you won't feel as much pain when you experience a fall, but wait till the next day and you'll feel that the pain is actually very excruciating. So I'll describe a little bit more about the opioid peptides. So they all share a similar N-terminus pattern, which is a combination of tyrosine, 2 glycines, and a phenylalanine, which we can see over here, and also on the structures over here, which surprisingly shares the same structure as morphine over here. But what happened is that with these opioid peptides, we believe that there is a potential allosteric interaction with the opioid receptors. So we can see over here, whereby this is just an introduction of the concept of allosteric modulation, whereby the common drug receptor pocket is called the allosteric site, where we will get the normal agonism. But what happens is that when we can alter the signal of the receptor based on attaching onto the allosteric site. And so we can actually manipulate the signal based on the receptors, based on the allosteric site receptor interaction. So for example, methamphetamine will actually has an extra structure on this end, whereby it can actually potentially interact. So we will move to dynorphin A, which is also a Kappa opioid receptor agonist. And so there is a message and address region within the opioid peptide. So the message region is still the same, and it shares how the similar structures to morphine, but there is an address domain which actually alters the selectivity of the receptor as well as the potency. So we can see from the opioid peptide, there are actually much more places to interact within the receptor. And thus we believe that it is a potential way of having less side effects. And this is observed throughout nature, whereby if a person who is injured, they won't feel the pain, but they won't get addicted to that endogenous peptide effect. If not, then we will just be more or less inflicting injuries on ourselves all the time. So this is how we actually use this as a base model, but this creates a problem. So remember that I was talking about how these opioid peptides only work for 1 day. This is because that this peptide is susceptible to physiological enzymes. So these are a list of physiological enzymes that are present within our body, but the main enemy, more or less what we call it, is trypsin. So trypsin actually cleaves between arginine and lysine amino acid groups, thus degrading the peptide and reducing the potency and efficacy of the endogenous peptides. So this is why we actually only feel the effect for 1 day. And then once we're in safety, you'll feel the pain right away. This is because most of the secretions from the immune cell has already finished. And at the same time, the endogenous opioids that have been secreted will be degraded by our physiological enzymes. So this is more or less the problem that we have with endogenous opioid peptides. So let's just jump out for a while and actually look into insulin, which is a very interesting peptide. It's also similar peptide. It also has arginines and also lysines within the structure, but you can see that this insulin is also more or less what we can say a druggable peptide. As you can see, there are many different types of insulins in the market, where there's rapid acting, regular or short acting, intermediate, and also long acting. So this is a model that we can emulate for the endogenous peptides that we have. So one of the few natural features in insulin that we have is actually whereby there is disulfide bonds, whereby it will create kinks and also create a specific shape out of it through cysteine interactions between the two peptides, and thus creating a more or less cyclic structure in the case of insulin. So we can take this further and actually make it much more stable by actually introducing amino acid substitutions, whereby we have salt bridges, where it is more or less an ionic interaction between lysine, glutamine, and also aspartic acid with arginine. So what we have over here are actually on the market insulin products called insulin glucinin and also gluten aspart. As you can see from the name, it actually hints the amino acid substitution that we can see over here. And so these amino acids will actually re-interact with the structure and start to fold itself to actually be much more stabler and less susceptible to enzymatic degradation. So the other way is also to use lipidation. So by using a long chain fatty acid over here attached to one of the amino acids, will actually create it to actually be more readily absorbed to the fatty tissue and also plasma protein binding. So it prevents itself from being discovered by the enzyme and then reduces the degradation and will be released over time as the concentration and the concentration decreases. More of the insulin would be released out from the plasma protein, and then this will actually create a long-term effect. So this will actually work on the long-lasting insulin. So how do we make a peptide? So this is where it goes to my project, where we are lucky enough to actually have a peptide synthesizer, whereby we have a reaction chamber in the middle over here, assisted with microwave to actually create some heating, and also an oscillatory chamber to actually mix the peptide amino acids. And so then over here, there are amino acid vials that we can actually dissolve, and the machine will allow to pick it up, conduct the reaction. So what reaction happens is essentially we actually synthesize based on a resin, whereby the amino acid, the amine group will be exposed into the resin, and we will actually run a series of reactions where we deprotect the resin, allowing the N-terminus to be exposed, and then we can actually attach it an amino acid that is protected on the other N-terminus, where the carboxyl group will bind together into a coupling reaction. So this creates a singular pattern as we synthesize our peptide. So it's a very efficient reaction, so long as the conditions are right. So after that, to ensure that our peptide is real and it is the product that we want, we will determine the mass through liquid chromatography mass spec to determine the mass. So as you can see over here, like this is an example of leuenkephalin, where we can see that the mass of leuenkephalin is 555, but we add a hydrogen because of the ionization effect, 556. So then that's our product. While we also may have side products within the crude, so we will actually then run a purity test and also purification using high performance liquid chromatography, whereby we actually let our molecules pass through a column where it will separate out depending on the properties, but we use hydrophobicity in this case, whereby the least hydrophobic components would come out first, and then slowly as we change the gradient, we will actually release our peptide. Then how do we actually test for stability? So in order for us to actually move forward with our peptide, we will actually have to test it in rat plasma, more or less similar to human plasma, where there are different enzymes present in there. And then we also expose it to a known enzyme, trypsin. And also, as we conduct our assays, we also use, and our assays also do contain certain proteins. We also will actually test it in assay medium, where we actually expose it to body temperature. And then we actually determine the mass again using the mass spec, but with a much more specific separatory methods. So by using a gradient, we can actually separate out the different peptides that are present, and from there we can determine where it cleaves our peptides. I do apologize in advance that I can't really show how it cleaves based on our research, but I can just show the methods that we have done. And the next is to assess the efficacy, which is through if you remember the explanation I had through the opioid peptides, cyclic AMP is released in the basal condition. So in this case, we actually quantify the amount of cyclic AMP using a cyclic AMP inhibition assay. So whereby we expose cells to a certain stimuli and it will start to secrete cyclic AMP. Then we add our opioid, novel opioids, to actually see if there is an effect. So in this way, we can see, in a way of translation, we can actually translate how much pain is being inhibited with our opioid peptides that we make. So using a positive control, we would use U50, which is a Kappa opioid receptor specific agonist, morphine as mu, and SNC80, which is also a delta opioid receptor agonist. So from here, it will actually compete with a biotinylated cyclic AMP tracer, whereby if there is less cyclic AMP, there will be more chance for the KIT cyclic AMP to bind and thus have a stronger emission. So we can see as we increase the concentration, we have a higher emission. While a lower concentration, you have less emission because more of the endogenous cyclic AMP would bind to the receptor bead. So I'll just briefly, so that's more or less the end of my talk. I'll just briefly talk about my lab just to explain how a PhD project can fit in. So as you can see, this project is quite big in the sense that there is the synthesis, there is the biology, and then also we will then intend to go into animal testing, in vivo models. So this is where we actually need that collaborative model between two labs where a marriage of biology and chemistry comes into play. So these are my two supervisors, Peter Cabot and Harry Perec. So Pete is my primary supervisor. He works on inflammation, pain, and biotransformation of our peptides, both in vitro and in vivo models like I've described earlier, while in vivo is mostly on pain models on animals, while my supervisor, Harry Perec, he specializes in chemistry, where he has multiple projects on drug delivery systems, whereby the Sol-Gel delivery system that I've mentioned briefly. And also you can see from the promotional slide, promotional material that was given. So this is basically an example of our Sol-Gel, whereby, let me just, oh no, it's not playing, is it? Oh, well, basically by applying the Sol-Gel directly into the warm nasal cast, it will then adhere to the roof of the nose. And this is where it won't be obstructed and slowly release to the brain over a period of 3 days. So we can use a lot of different drugs for sustained release. And this is also where we can also mix and match from the peptide synthesis into sustained release models. So a little bit of appreciation. My lab, all my friends in the School of Pharmacy and also in UQ, as well as the friends in my life and also my partner, Lexi. And then I'll just close it with a remark of that in order to actually be inspired, sometimes looking through nature helps a lot. So for an example, an hypodermic needle that we've used to deliver the vaccine, it is actually more or less taken from multiple venomous animals that have the same injection kind of mechanism, as you can see over here. And it is a very interesting thing that we actually emulate it from animals without actually thinking about it. And finally, pharmacology is benefited by the prepared mind. You need to know what you're looking for. So we know that we want to curb opioid addiction. So we would find tests to prevent that. And thank you very much. I will accept any questions that come up on the floor. Thank you so much for the opportunity for sharing.
Yeah, so thank you to our speaker for tonight, Danial Saifuddin. That's such a very informative presentation. And we really know that this opioid actually is a big issue because few years ago they have this so-called opioid crisis in the US, and it caused a lot of issues. That's a big issue. So
Yeah.
we have several questions from the audience,
Mhm.
and this one came from Ying Xianko, and he asked,
Yeah.
different people have different level of self-generated opioid peptides since different people have different sensitivity towards the pain. So,
So yeah, thanks, Yingxian. That's a very interesting question, actually. What I would say is that it really depends on the person's immunity in the sense of the immune cell response, because this solely lies onto the macrophages and also the neutrophils within the body to actually secrete out these opioid peptides. So people will have different levels of opioid peptides that are generated, but what we can see in the trend, and this is more or less in the wide scheme of things, most people actually does produce opioid peptides. It's just how much, and usually it's enough to actually prevent the pain from being sufferable, to the sense that they can still move to safety or apply first aid before it gets worse. Yep. I think, do you want me to read the next question, or Atika?
I
Yeah.
I can actually read it. So,
Yeah, thank
is
you.
it something to stimulate the production of opioid peptides, or the opioid peptide is generated in a homeostatic fashion in our body?
So this is actually also another interesting question. It's actually the inflammatory factors within our body. So it depends on whether we how much inflammation we have, then we actually the immune cells will actually start secreting that amount of opioid peptides. But it's usually just enough to curb the pain. But this actually opens up a very interesting point of view where some people who have constant inflammation, will they actually feel more or less like sedated? But it doesn't. This is where it is an interesting point, whereby we don't get that sedation with these opioid peptides. So this is where it is an interesting point of view of using endogenous opioid peptides as a drug. But my project is to focus on creating this endogenous opioid peptides as a drug later on, in more or less like in the clinical environment. But yeah, that's the more or less the stimulation of production is based on the amount of anti-inflammatory, the inflammatory factors that are secreted through the inflammation. Usually, it's an injury. Yeah.
Okay, so come to the
So okay, so how do we end? I'll go to the next question. So how do we ensure that the opioid peptide is efficacious and or functional, and at the same time not addictive? This is an interesting question. I really so how we would first determine the efficacy of our peptide is through the cyclic AMP assay. And this is where I'll actually cycle back my slides to the other mechanisms. And also there are other tests that we can perform for addiction and also tolerance. Give me a second. Yeah, so initially about efficacy, I've mainly focused on GPCR signaling, whereby the cyclic AMP would be released if there is no opioid peptide. But in the event of addiction and tolerance, we would look into beta-arrestin signaling, whereby how much of the receptors are internalized. And there is actually a few cool tests to actually determine that. Some is actually directly measuring the secondary messengers such as PERK or the P38, which they are kits to determine the amount of it being released. But the other interesting thing that we can do is literally antibody, like tagging the receptors with antibody and then a fluorescence antibody, whereby then it will start to fluoresce. So from there, we can actually make a temporal view of how the cell receptor internalize when we apply the opioid peptide. But I don't have any images that I can show at the moment. So you can actually look up immunofluorescence of opioid peptides on the internet and for internalization, and you can actually find a lot of cool pictures where if a receptor is within the surface of the cell, you can see that it is around the surface of the cell. But at the same time, when it's actually internalizing, you can actually see the fluorescence inside, literally like inside the cell, where it is focused into that particular region. So these are actually some ways to actually determine the tolerance of our peptides. Let me just double check the question.
So we have
yes.
question from Facebook.
Yep.
Yeah, so this one is from Chin. He said that, good evening. Does the cocaine drug have a similar mechanism as the opioid do?
Can you please repeat the question?
Does the cocaine drug have the similar mechanism as the opioid?
Interesting. So yes, cocaine is actually used as an anesthetic in the clinical setting. And it is actually to numb like the different receptors, other receptors within the opioid. But because more or less, cocaine is a very dirty drug in what we call it. So it does hit a lot of different receptors, and opioid receptors are one of the things that they hit. And so thus there is that anti-nociceptive effect with cocaine, but there is also a heap of other things that it hits within the brain. And this is why it's often used for ear, nose, and throat operations. But at the same time, it has to be very carefully administered, because if it hits the brain, there is a heap of receptors such as GABA, and it will hit all of these receptors, which will create a problem for cocaine. And this is why we don't really use that as a model for pain. But yes, it's a very dirty drug in the sense that it will hit a lot of other receptors. Yeah.
Okay, so the last one is that, so if we produce the opioid peptides in our body, right? So then we actually don't have the opioid drug supplied by the pharmaceutical company, or how?
So yes, this actually goes back to the point within my presentation. Yes, we do produce opioid peptides, but they are degraded very easily. So we do feel pain for a short. We do have that opioid peptide effect where we don't feel the pain for a short while, but later on, there will be that pain effect because these opioid peptides are just that unstable. And this is why we are looking for ways to stabilize these opioid peptides. Right? Does that answer the question?
Yeah.
Thank you.
Okay,
Thank you for the questions. It's a very nice and interesting discussion to have. And I really like that we get everyone's brains turning for the different perspective of endogenous opioid peptides. Yeah.